Hello and welcome to the Practical Animal channel. The channel is for you if you want to know what it takes to work with animals. That could be with domestic species, companion animals such as dogs and cats, or it could be with farm animals. Or perhaps you want to work with wild animals, either in the wild, such as a field naturalist, or in captivity, as a zookeeper, for instance. The channel has its origins in bird of prey training and falconry, and I'd like to say thank you to one of my most consistently loyal subscribers, Pete, and his channel, Free Bird of Reason, for mentioning my channel recently. Anybody wanting to know how to train and hunt with a Harris's hawk in a really in-depth way, and many would say that that is the only way, needs to see his channel. This week we continue with the sixth and final episode in the series of interviews with Professor Mark O Shea, MBE. Mark is a Professor of Herpetology at the University of Wolverhampton in the UK. He is also the former presenter of several high-profile TV natural history documentaries, predominantly on snakes. Following on from the previous episode, in which I'd asked Mark about what is involved in describing a new species of snake, in this episode I ask him the follow-up question. Presumably the conclusion of any paper describing a new species of snake is going to include a recommendation to conduct a survey to see if the species is critically endangered or not.
That could quite possibly be the case, and it certainly could be the case with, and this could apply equally to birds and mammals, that, a wide-ranging, say you've got a wide-ranging species that's found right across Africa from Senegal to Kenya or Somalia. And it's found all the way across in a number of different habitats. But as you go across, it looks the same, but it might actually be a species complex. There's one in Britain, and I'm not an ornithologist, so please forgive me, but there are two species of gulls that occur in Britain, and I should really look up what they are. I think one might be, I'm not going to put my foot in it. There's two species of gulls that when they're in Britain, they look very different. But if you start going, they overlap, but their ranges end sort of in this area. And one goes eastward and one goes westward. And if you follow them round, eventually they become basically the same. It's like it's called a Rassenkreis, a ring species. And you can have things like that, where at the two ends of the extremes, they look different. But in actual fact, as you go through the population, you see that it actually looks like it's the same one. But if you could equally have one wide-ranging species, and it occurs on all sorts of different habitats. And when you start to look at it in detail, certainly with the molecular techniques available to taxonomists today, you find that it's actually 5 or 6 species. And some of them may have very small ranges that you then feel that you should be trying to protect. And so that's when it becomes very important to actually take action. I think the St Kilda wren in Scotland is a subspecies of the wren we find in the rest of Britain. Again, the ornithologist will be throwing bricks at me because I'm not an ornithologist. But if it's is it troglodytes? Troglodytes is the species throughout the whole of Britain, and then the one on St Kilda is a subspecies. Troglodytes St Kildarensis, for sake of argument. Well, that would say this is a population that requires protection. Ornithologists, please forgive me. The taxonomy of birds is not my area, but that's an example. And again, an island form. And island populations are obviously those that are often threatened, because once they're gone, they're gone. It's not like they're coming back in across the border. So island populations are vulnerable, yes.
And within the context of your best snake fact, the final question on that subject is, you've mentioned terms such as biodiversity exploration, holotype, paratype. Can you just explain those terms? And also,
well, biodiversity explorations speaks for itself. You know, we are investigating and exploring the biodiversity of the planet. Each taxonomist or biogeographer or whatever is looking at their particular taxa, the group of animals that they're familiar with, and determining how many species there are. And we are forever, as I've said, raising the numbers as we discover new ones. So the exploration is quite obvious, really. You know, we're going out there and we're looking at these animals, and it's super important, because in the past people didn't look at things they weren't interested in. Reptiles and amphibians and spiders and various other groups weren't as popular as birds, butterflies and beetles. And so all the work was done on birds, butterflies and beetles and mammals and things like that. You know, but other groups have got their enthusiasts as well. As for holotype, paratype, holotype is the specimen that you use to actually describe your new species, and you will describe that specimen in great detail. The paratypes are other specimens collected at the same time in the same locality or very close to the same locality that support the holotype, and so then you would document the variation. They would provide you with ranges in scale counts, because you'll only get one scale count for the ventral scales from your holotype, but if you've got 3 or 4 specimens, then you'll get other ventral scale counts that'll show you what the range is in that species or from those individual animals. And again, obviously, as more specimens are found, you might go into a museum and find, for instance, we described a new species of ground snake from Papua New Guinea last year, and my colleague he collected the specimens. There were 3 of them in the type series, a holotype and 2 paratypes. And when I looked at them, I remembered I'd seen something very similar. His were in the South Australian Museum, but I remembered I'd seen something very similar in the Australian Museum in Sydney. And so when I went back over, I photographed and documented that specimen, checked where it had been collected, and yes, it had been caught very close to where his 3 specimens came from. So in a museum, I found another paratype, and existed. And the point is, it had just gone in as a stegonotus. They're such a complicated group that nobody felt like looking at and working out what it was before, but it jumped out at me as I recognized the same characteristics. And it was, I mean, this was a very white animal with a dark head. It's quite spectacular, a nocturnal snake that almost glows when you see it in the dark. But it was very, very interesting to find another specimen that in another collection of, and there could be other specimens from this species sitting in collections as yet undiscovered, if you like. And when people turn them up, they go looking through a big jar of stegonotus, and they'll find one. They go, well, hang on a minute, that looks like that one that O'Shea and Richards described. And they dig it out and they'll do the scale counts and look at where it was collected. And I go, yeah, it is. Okay, we've got a stegonotus apple in here as well.
I know I said that was going to be my final question on that context, but just tell me, Mark, and bring this subject alive for viewers, please. When biologists consider museum specimens, is there only a finite usage within biology for those species, or is there an
no? There's other use. If you want to know what's happening to a species over time, looking at museum specimens are very useful. Say there's a big collection of a particular, and I'll just say a particular kind of snake, particular species of snake. There's a big collection in museums around the world, and it's been collected all over its range for 100 years. And there's in museums around the world, 150 specimens. If you are looking at those specimens, there's some from the 19th century, there's some from the early 20th century, there's some from the late 20th century, and there might have been a few collected recently. And if you're looking at them and you actually open up their guts of the preserved specimens, you sometimes find what they've eaten. And so you might find that 100 years ago, they were feeding on a native mouse. But the recent specimens are feeding on an introduced mouse that's pushed the native mouse out. They may have changed, and they may, that if you look at the reproductive state of the females, and the males, the size of the testes, but the females especially, you might find that only over a certain size were reproductively active, developing eggs 100 years ago. But females now are smaller to reproduce. And that could be because of other pressures on them. They're not growing as big as they used to. And so in actual fact, when you've got a lot of specimens like that, you can look back into the past. They've done it with chytrid. Chytrid's this terrible fungal disease that's affecting amphibians, and it's killing them around the world. And they've been able to trace back specimens from the 1950s, I think it is, that have got chytrid spores and things like that. So you can start looking for diseases and go back in time. And so literally natural history museums are time machines. Not only are they treasure houses, they're time machines. They encapsulate the history of that species over a long period of time and enable you to answer some questions. So yeah, they've got a lot of value beyond just working out what biodiversity is on the planet.
Mark, is there anything that you'd like to add?
Off the top of my, I mean, when I'm normally asked that, I normally say, document any unusual behavior you see and write it up as papers, but I've already said that. And the other thing I say is don't lose sight of your goals, and I've already said that. So I'm a bit at a loss of another sage piece of advice to deliver at the moment.
If I can ask a question then, just for a change, would you say that there's any sort of time limit to publishing a short note on something that you think is worth publishing? I mean,
not really, but if somebody else publishes it before you because they found the same thing, then probably, isn't as relevant. We had a parthenogenetic birth at the safari park, and one of our female anacondas, and there's only one parthenogenetic is when it's this female reproduces without the need of a male. And there are a number of obligate parthenogenetic species around the world. There's quite a few lizards. Some of those are the product of a hybridization event between two sexual species. There's only one snake, the Brahminy blind snake, tiny little snake that most people don't recognize as a snake, and travel around the world because it turns up somewhere in the root ball of a plant and it's nice and warm. She starts a family. She doesn't need to lonely hearts or anything. That's an obligate parthenogen, but there's also a facultative parthenogen, which is when a normally sexual species in the absence of a male reproduces. And our green anaconda, one of our green anacondas, we only kept females because we weren't trying to breed them. One of them gave birth, and she gave birth to 8 offspring. That's a small litter. That's a sign of facultative parthenogenesis. 5 of the 8 were dead. That's another high mortality. That's another sign of facultative parthenogenesis. They're all female. That's a sign of facultative parthenogenesis in pythons and boas and anacondas and things like, whereas they're all males in garter snakes and rattlesnakes. Anyway, it was worthy of publication. So I wrote the paper up with a couple of my colleagues, some of my colleagues from the safari park. We got that note published. It was the first instance because I'd researched whether it had been recorded before, and it hadn't been for green anacondas. It's not, sometime after we'd published, somebody contacted me from South America, and they said, we had that happen in one of our collections. Well, you should have published it. because if you'd published it, ours wouldn't have been so important to note. We might have said, here's a second instance, but they didn't publish it. Therefore, as far as science is concerned, it didn't happen. So if you've discovered something, don't sit on your hands. Otherwise, you'll end up like Darwin almost did with Alfred Russel Wallace, sending him the same idea that he'd been sitting on for 20 years and saying, would you present this at the Linnaean Society for me, please? You know, oh my God, that's my idea. Nearly missed out. He waited 20 years. So don't wait 20 years to publish it. Publish it as soon as you can. But you probably need to, might need help from somebody who's used to writing those papers and a co-authored paper.
Professor Mark O Shea, MBE, Professor of Herpetology at the University of Wolverhampton. Thank you very much for being on the Practical Animal channel.
Thank you very much. It's been my great pleasure.
That's the second series that I've uploaded onto the channel. The first was about a year ago, a two-part series on how to bond with your Harris's hawk. Mark was really interesting to speak with, and I hope you'll agree it was worth making his interview into a 6-part series. The actual interview itself took place over 2 days and was a total of 2 hours. If you know anybody who works with animals professionally, who might be interested in being interviewed, please forward this video to them. If you liked this video and found it informative, please consider clicking like. And for more exclusive content on nature conservation and the animal industry, please consider subscribing to the channel. Coming up in the next 3 weeks, we have a person who is an elephant conservationist. He has been a TED speaker and he advises zoos and other facilities on best practice in elephant management. I also speak to a bird scientist about what it takes to be a professional ornithologist and research scientist. And we talk once again with Jemima Parry-Jones about her decision to close the International Centre for Birds of Prey to the public and what the future has in store for her. You're not going to want to miss that one. In the meantime, have a great weekend, and until next time, thank you very much for your time.